Hello everyone, thank you for clicking on the Literacy Volunteers of Harrison County YouTube channel. We're a nonprofit United Way agency. If you would like to subscribe to our videos, please click on the subscribe below. If you hit the bell icon, you'll be notified every time we put out a new video. This is the last on my series of genetics with biology, and then we're going to start with the fourth subject on the next biology lesson. But this one is lesson 7 on epigenetics. We talked about DNA and DNA structure, and then we looked at genes, and so now we're going to look at epigenetics. But before we discuss epigenetics, we have to define a couple of terms so that you aren't too confused when we get to the topic. There are two different types when we look at our genes. You can look at your genotype, and that's the actual genetic characteristics of an organism. What gene you have. If you have blue eyes, you have a gene for that. If you have brown eyes, you have a gene for that. That's your genotype. The phenotype is the physical characteristics of that organism. So you have a gene for blue eyes. Do you have blue eyes? That's the physical characteristic. That's the phenotype. And we can look at that a few ways, and this is the way we typically do it in general biology. We're looking at genes. Big B is a gene for brown eyes. So big B is the gene for brown eyes, and little b is the gene for blue eyes. When we have a big B and a little b, this big B is said to be dominant. So brown is dominant over blue. So if you're big B, little b, and we call this heterozygous for brown eyes, then you are brown eyes. If you have big B, you also have brown eyes. The only way to get blue eyes is to get little b, and then you have two recessive genes. This is called homozygous for blue, and you have blue eyes. So brown eyes, blue eyes. The genotype is homozygous blue. The phenotype is blue eyes. So we have to kind of go over that before we look at epigenetics, because if not, then you'll be lost. So there we are. There's our dominant, is the gene that gets expressed. So if you may have the gene for blue eyes. Okay, but if you have the gene for brown eyes, you're not going to have blue eyes. The brown eyes are going to show up. That's why two brown-eyed people cannot give birth to a blue-eyed baby unless they are heterozygous for brown. And so here we're looking at the recessive gene only shows expression when you have two copies of it, little b. And so epigenetics, when we start to look at those, epi, the prefix means upon or over, and of course genetics, your genes or your DNA. So these are things on top of your DNA, and we're going to look at those characteristics. So the field of epigenetics studies the changes in organisms caused by modifications of gene expression, rather than alteration of the genetic code itself. We're not changing, we looked at the A's, T's, C's, and G's. We're not changing your genes. We're looking at how they are modified by things that are added on top of or onto those genes. So here we look at what is a mutation. You've heard of people like the Mutant Ninja Turtles. What is a mutation? A mutation is a change in the genetic code. We're changing those A's, T's, C's, and G's, right? A change in the DNA sequence, the actual letters that we looked at twice in the previous two videos. If we have a germline mutation, that's a mutation that occurs in either an egg or a sperm, and when those come together, those are passed on. Those are passed on down the line. Color blindness is passed on, right? And if we look at somatic mutations, soma means body. So all cells that aren't eggs or sperm are called somatic cells. And so those occur in body cells and they're not passed on. Say you're already you and we change one of those genes mutates, you can't pass that on to your children. Only if it's a change in a germline cell, right? So these mutations are caused by mistakes when the DNA is copied. We looked at that last week. It's called translation and transcription, right? So when your DNA copies itself, if it makes a mistake, then it's going to keep passing that on. You also have a check where your body will go, oh, that's a mistake, and get rid of that DNA. So, but sometimes it stays. And those are result of environmental factors. We have UV radiation. That's why they tell you to watch out if you lay out in the sun. Diet, stress, cigarette smoke, drugs, all of those things can cause genetic mutations. So while the sequence of the DNA may not be affected by your environment, right? The actual A's, T's, C's, and G's, the way genes are expressed can be, and that's what epigenetics is. And so it's a brand new, sort of since 2000, we'll look at how it came to be. But our DNA, we've already looked at it, is in your cell, and when it's all comes up like that, it's called chromatin. It's wrapped around these histone proteins. Here are these histones. So you have a protein and all this DNA is coiled tightly around it. And then on little pieces of the DNA, we have these little flags. We can have an acetyl group or we can have a methyl group. And those groups determine how tightly wound around that histone protein and how much we let it go. Once we let it go, we can make a copy of it. So we're governing the DNA by how tightly wound or how loose it is around these histone proteins. So we can only achieve gene expression when we unwrap the DNA, right? So now we're expressing our genes to see what makes you, you. These methyl groups, acetyl groups, and these little histone tails that we see on here affect the way the DNA is read and how tightly it is bound to the histone proteins. So we can either turn genes off or we can turn them back on, right? So those are the ones we talked about that are going to achieve expression. So what causes these changes are environmental factors. We just looked at that. Food, drugs, cigarette smoke, UV light. That happens, that changes how those methyl groups appear and don't appear, or those acetyl groups on your DNA, and then it can change what happens to you and the effects of that. So we have two different effects. We can get a slight change in gene activity, which is not that bad of an effect, or we can get dramatic changes by switching the genes on when they should be off, or vice versa, which leads to disease. So these changes are heritable, which means they can be passed down, right, from parent cells to daughter cells, from parents to offspring. But we want to know, how was this discovered? Who discovered epigenetics, right? So in the year 2000, we have two scientists at Duke University, Jerdal and Waterland were their last names, and they designed this groundbreaking experiment. They started with these what are called agouti mice. This is what this agouti is. And agouti is a gene that makes these mice yellow. They have yellow fur, and they have what's an insatiable appetite. They eat and eat and eat and eat, so they're super fat and they're yellow, and the gene also makes them more prone to cancer and diabetes because of that agouti gene. So what they decided to do was to take these mice before conception, and they fed the mothers of these mice, a test group, a diet that was rich in these methyl groups that we just talked about, these donors, these CH3 groups. And so they gave them garlics, onions, beets, kale. Those are the things that add methyl groups to your DNA. And then they, this was before they were conceived, and then they had the mice. The resulting mice looked different. These mice now are skinny, slender, and brown. They're not fat and yellow, and also they go back and look and they don't have that cancer diabetes gene link. Okay, so the thing to remember is they still have the same genetic code. All we changed were the amount of methyl groups on their DNA. We added methyl groups. So the results were the effects of the agouti gene had been erased, right? So what can we conclude by this experiment? Instead of changing the mother's DNA, we only changed her diet. And what we did was add these methyl groups, and they worked their way into the genes, remember, that are around the histone proteins in the chromosomes of the embryos and onto this agouti gene and turned it off. Remember, we're not mutating. We're not changing the genetic code. We're adding methyl groups to turn that gene off. And once it's turned off, it no longer achieves expression. We looked at genetic expression. So now we have these mice that look totally different, but still have the same exact DNA that their parents had, right? The mothers had still passed the agouti gene on, but we're not expressing it anymore. We've turned that off. The babies have it, but we've turned it off. But thanks to the methyl-rich diet, they added the gene and a chemical switch that dimmed or caused those effects, those deleterious effects. So they've done a lot of research on this. They also did research on mothers who were pregnant during 9/11 when they bombed the Twin Towers, and then they went and checked the babies afterwards. The high amount of stress that they went under negatively affect the babies and gave them anxiety and different things like that. So what we're looking at is not only are you by your parents, your mom's and your dad's DNA, but things have been passed along to you by your grandparents and your great-grandparents. These methyl groups and acetyl groups groups and how well you adapt to those things. You can change your DNA now. If you eat those methyl groups and you methylate your DNA, you will become much healthier, and we can work on turning genes off and turning genes on. So if you liked this video on epigenetics, please give it a big thumbs up, share it, and subscribe to our channel. Thank you.